Ask them again if they can hear me. Well, if they're not replying, they can't hear me, so.
Teresa, can you hear Sharon?
Sometimes.
Yeah, I'm like Dick sometimes.
They're saying only sometimes.
Okay, let me stand here for just a second.
This is Patsy, and I'm hearing her all the time.
Thank you. I know, I'm bouncing because I'm nervous. Welcome to the April 2023 Cross Timbers Master Naturalist Chapter meeting. I'm talking through, is this your computer, Breena? I'm talking through a computer and it'll be a minute because we have a few announcements. Technology is wonderful when four of us can figure out how to work it. So thank you all for being patient with us. And hello, everyone. I want to start out by saying happy National Volunteer Week. Pat yourselves on the back. Yes, and I know we're busy this month, so make sure those folks at the sponsoring groups where you're going, River Legacy, Fort Worth Nature Center, make sure they know, hey, it's National Volunteer Week and here we are, so they'll at least realize we know. I don't want to unmute. Okay, thank you. I thought I told it to unmute this computer later. Okay. Another thing, just a couple of announcements I want to make. If you would like to take some online training sponsored by Texas Parks and Wildlife Department, you may or may not have gotten an email sent from Michelle Haggerty. If you're on the listserv for the state, you got that email. It's one of those, the road to hell is paved with good intentions. And this afternoon, I started to blast it to all the active members. And 5 emails later, I had to get ready to come here. So I will blast it tomorrow. It's Thursday from 7 to 8 p.m., April 20th. And it is training on the City Nature Challenge, which is coming up at the end of the month into the first couple of days in May. So I'll do my best. And if I forget, just email Sharon at president@ctmn.org and say, where's that message you said you were going to send us? There's also, if you're interested in the shirts and so forth that the state sells every once in a while, they've got their pop-up open, and I'll send you that link also. It's on the state website, and they're selling a few shirts and so forth. That has to be done through the 23rd. I think I have a really important announcement to make, and I apologize that I haven't pulled up all the particulars. It may be that someone could help us with that. But the important announcement, I think we'll all be glad to know about, is that thanks to our fellow NIPSOT chapter, and thanks, I believe, to Kim Conroe, although there may have been some other people involved, who talked to the people who are now running the Botanic Garden and said, hey, why are you planting invasives? There are some plans, although, as you know, with anything, it takes 5 years when it ought to take 2 weeks. There are plans to remove, I believe, the Nandina. Avon, do you happen to know? I'm looking at you because I'm looking at you.
I believe Nandina and Privet is also.
Nandina and Privet, but it'll be several years, and so if you get an email from either NIPSOT or from us here at CTMN that we can help pull those suckers out of the ground, you can think of that as all your old bad bosses. Every one of them you pull out of the ground. then just be aware of that. But it's going to be a slow process because, of course, they have to budget for replacements and so forth. But I thought, yay, they listened to someone. Yay. And that's all my announcements. So apologize, Breena, I didn't pull up your bio, but theoretically they all read it. Breena Davis, Breena Riley, Courtney Davis, Breena, come on up here, is graciously agreed to talk to us today about diatoms. And remember, if you're breathing, thank a diatom. I know it's true about plants too, but, and please go ahead.
I'm gonna get my water because just like a diatom, I need water as well.
Oh, that is the. Okay.
So I'm going to give a really brief introduction about myself. I kind of just fell into diatoms by in grad school. I went in with the intention of studying native plants. I have a background in stream ecology, and they're like, well, we don't have a space for plants. We do have a place for algae and diatoms though. So I looked into it. And I'm like, sure, primary producers, I'll go for it. And I came out knowing about algae and not anything about plants. So here I am today. Let's learn about some diatoms. Actually, I do know about plants, but I didn't learn about it in school. All right, diatoms, living crystals. So a little bit background about this slide. I love showing it because it shows the beauty of diatoms. This is from one of the studies that I did for one of my projects, but I think it's really great. It kind of reminds me of a kaleidoscope. So that's my background. A little bit about diatoms. They were first discovered they're microscopic in size. So diatoms weren't really first described and discovered until after the invention of the microscope. The first diatoms were described by one Otto Friedrich Mueller in the late 1700s. And about 100 years later, about 100 years later from that date in the 1880s and the 1890s, Victorian people had a habit of and a hobby of mounting diatoms and arranging them artfully. How they did that, who knows, but it's quite a talent, onto microscope slides, and they'd have viewing parties of okay. I apologize. Okay, let's try this again. If I have to do it with this view, then I will. So. Are y'all getting the? Okay, I think I know what's going on. So just like field work, you have to improvise. To take my samples out in the field, I had to use pretty much what amounted to a straw. Okay, so I'm just going to share it like this. Does this work out for everyone? Okay, so improvisation, again with the straw, had to suck down to the dirt, put a bottom on, anyway, I can describe that later. Anyway, so going back for the people who didn't see this slide, again, people would, Victorian people would trade these like trading cards. And it was like, gotta catch them all with all your diatoms. So going from the 1880s and 90s to today, we've learned a lot in the ensuing 100 years. So what exactly is a diatom? So getting into the nitty gritty, they're eukaryotes, so they have a nucleus, like you and I, and getting down to a few levels at the phylum level, they're actually distantly related to kelp. So when you think of like the West Coast and the kelp forests, diatoms are actually related to them. And then they get down to their own class called Bacillariophyacea. So what actually makes a diatom? So diatomos is from the Greek for cut in half. And this is because diatoms have a top and a bottom shell, kind of like a Christmas box. The shell is made from silica. It's it's a non-organic material, and that's what puts them apart from other types of algae. The other algae cell wall is organic, but the inorganic shell of diatoms doesn't degrade over time. So hence, it's a source of diatomaceous earth or fossilized diatoms. That's where you get your diatomaceous earth for gardening. They're also, another common name is golden brown algae. This is because they use a gold pigment or a gold color instead of green for photosynthesis. And this just has to do with an adaptation of them being in the water, so they use different wavelengths of light versus what plants do. So diatoms are pretty much found all over the world. So you can go even in the driest, deadest parts of Antarctica, you'll find a diatom all the way to the tropics. You can go from the oceans all the way inland. So from the seas to the deserts, anywhere there's water, there's probably a diatom. Diatoms are important for us because they're the base of many food chains in the water. In the ocean, for example, I believe that a number I read is that they make up like 60% of like the green material in the ocean. So you get rid of diatoms and you really start messing with the food web. But diatoms haven't always been around. They first evolved about 150 to 200 million years ago, which would be about the time of the Jurassic, with the dinosaurs is when they appeared. And then they started really exploding in population right after the extinction of the dinosaurs about 50 to 60 million years ago. So what is their general place in the food web? So again, they are microscopic in size, so we didn't really know about them until the discovery of the microscope. But they're primary producers, or the bottom of the food chain, the first level of the food chain, like plants, or the base of the aquatic food web. Actually, here on this web, it's not so much as a bottom of a pyramid as in a larger piece of the puzzle. But as you can see in this food web, if you take out any of these parts, like say the fish or the decomposers or the nutrients or the bacteria, then if you're missing a piece of the puzzle, that can really mess with your ecosystem. And diatoms are really important for us is because they can tell us what kind of changes, give us a clue of what kind of changes are happening in your water, with them being at the bottom, with them being primary producers or getting nutrients and food straight from the water, are really sensitive to change. So the diatom you see yesterday versus tomorrow, that change can tell you kind of, is there a change in temperature, or is there a change in nitrogen or fertilizer input to your water? So how do you tell if there are diatoms in your water? So I found all these pictures online. On the far right, you can see the aquarium water is tinted gold. And that's because of the diatom, I'm not going to say infestation, but I guess an algae bloom within this person's tank. They haven't been cleaning it properly. And you get problems in your aquarium when not everything's balanced. So this is a great example of an unbalanced ecosystem in a shell. How else do you tell in nature if diatoms are present? Well, just pick up a rock and oftentimes they feel slimy. That's probably from diatoms and the mucus that they produce, because they will stick to that rock. They're sticking to that surface so that they can have a place to sit and grab that sunlight. So a little bit about life history, or just how they live their lives. So diatoms, I think, are pretty cool. So like they produce so plants produce asexually and sexually, depending on if you get a cutting of them or if you grow them from seed. So diatoms are kind of the same way. Right here, they will, like bacteria, they'll produce from one parent and the parent will divide into two. But what's interesting is that over the generations, they will actually get smaller until they get so small they can't divide anymore. And then they switch to sexual reproduction, which will create a really big cell again. And what we see here is that each divided cell gets 1/2 of the shell, and then they'll make a little bottom shell in the bottom. And that's why it keeps on getting small. That bottom shell keeps on getting smaller to a certain point to where it can't divide anymore. Where do you see diatoms? Where do they grow? So they have all sorts of strategies or ways that they habitats that they fill. So I think one of the most fun places you can find a diatom is growing on a turtle. It's a hard surface. The turtle shell is facing the light. So this is specifically green algae on a common snapping turtle I saw, but I will guarantee you that there are diatoms somewhere on that shell. They'll also grow on other plants and other algae, anywhere to where they can stick. Also, sometimes you'll see them moving. They'll live in sand or they'll live on the rocks, and they move. Some of them move so that they can find a better place, like a plant will move to face the sun to get a better position in the sunlight. Or they'll float. That's called planktonic algae, or plankton is whenever you have your floating algae. So just like anything else in nature, diatoms have certain pressures or certain things that they have to worry about when they're growing in nature. Just like you have a place to where you may not like the hot summers, or you may not like the really cold winters. You have your happy place. Diatoms have their happy places too. So 6 areas that they have their happy place they want to find is with light, nutrients or food, water speed and depth, temperature, the surfaces they grow on, like that turtle or another plant, and pH, which would be how acidic like vinegar or how basic like bleach it is. So 4 things we're going to focus on are nutrients or food, water speed and depth, the temperature, and the growing surfaces. So let's start with the nutrients. So you have two conditions in water. Typically, it's on a spectrum. So you can have really high nutrients or high amounts of food, or really low, which would be eutrophic, or really low amounts of food in your water, which is called oligotrophic. So diatoms, with them being photosynthetic or producing their own energy, being primary producers like plants, they have similar energy needs or similar nutrient needs. Nitrate, when you look on your fertilizer bag, you have the NPK, or the nitrogen, potassium, and phosphorus. Nitrogen is for proteins and DNA production, and phosphorus and potassium help with your photosynthesis. The one special thing about diatoms is that they need silicone as well to make that outer shell. And they also, for sugar production, they need, I include sunlight as a nutrient because they need it to make food, and they also need carbon dioxide. Where do you get the nutrients? So nutrients, when we think of like pollution in water or rain, or when it rains, all that stuff from the land is entering the water whenever it rains. And that's why they always tell you clean up your oil. You don't want a lot of concrete because it'll wash fertilizers and oil into the river, and that stuff's not necessarily good for diatoms. So land use is a big thing. Whether it's farming, might put in certain fertilizers, which will put the food out of balance. It's like having a bunch of cookies instead of salads. Or if you have like a forest or mountains that are natural, then you'll have more of those salads versus those cookies. Temperature is also really important for diatoms. Some diatoms really like it cold, and some of them like it really hot, like the ones you'll find in summer around here. And temperature is also important because it affects the growing seasons. Just like plants, diatoms also have growing seasons. You'll have different seasons for spring, different species that'll grow in spring and in fall around here because of the temperatures. And as I said, some species only grow in spring versus fall because that has to do with like different rainfall and the temperature. An interesting thing about like how hot it is, the hotter it gets, the less oxygen or air that the water holds. And this is important when it comes for algal blooms and fish kills, because now, while diatoms use carbon dioxide, they also use oxygen like other eukaryotes, like us and plants. So they're competing for oxygen with the fish when there's not much when it's hot. And that's why it becomes a problem whenever it gets too hot. Some of the surfaces they'll grow on, again, non-living and living. Non-living will include like mud or sand and rocks. Living, I think it's kind of funny, algae will grow on algae. So on the left, again, we got the snapping turtle. They'll grow on turtle shells. And the one on the far right, those long sticks are the diatoms. That's called onaria. And they're growing on a green filamentous algae or green stringy algae, which is what you are likely seeing on that, what you can see with the naked eye on the turtle. So water speed and depth, or how fast the water is flowing, is important too. Think of if you're like in a running river. If the water flow is too fast, you'll have a hard time holding onto those rocks and you could get washed away. So that's a concern for diatoms too, because they don't want to get washed away. So you have different environments that you might find out in nature, whether it's river rapids on the left, shallow, gentle creek in the middle, or flooding conditions where the water gets high and you might have a lot of nutrients or food entering the system on the far right. Water depth, how deep the water is, affects how much sunlight is available. So you may find different species depending on that, like it deeper or shallower. And then water current is important because like for fish, it'll help bring the air in and it'll help bring the nutrients in, but you don't want too fast to water because again, you don't want to wash away your diatoms. So again, are there diatoms in my water? So what are some signs? So texture on a rock. So on here, you can see kind of, this is at a park, at a local park, and there's some rocks with some algae in the bottom. So you can look down, and if you see fuzz on the rocks in the water, then it's probably green algae of some sort, but there's going to be diatoms growing on that. Again, texture, it'll feel slimy when you pick up that rock or that turtle shell. Visual, it'll be kind of shiny. And going back to texture, it's like if you've ever been to, like, say, Turner Falls, and you have to be careful walking on the rocks because it's really slippery. Thanks, diatoms. And then visual, algae on rocks. And then where would you find them? So you can find them, again, if you were to look under a microscope, you can find them floating in the water, or you can find them, take a scraping and you can see what's scraped the rock with a toothbrush, which is how they actually sample, and see what was growing on the rocks and the plants under a microscope as well. So shell structure. So I bring up the topic of shell structure particularly because it's important when you're wanting to ID them or key them like plants. A lot of the species are described by their shape, and the ridges or the ribs and stuff on the actual shell. So if you were to look at an actual, if you were to look at an actual diatom under the microscope, this is what you might see like living. This species is called Navicula, or this genus is called Navicula, and it's growing on green algae. So let's describe the shape a little bit. It's kind of a diamond. The two gold lines on the edges are where your chlorophyll is, the golden chlorophyll. It's called fucoxanthin. That's that's the color that it uses for capturing sunlight. Some other things you might see when you're looking at it, but you can't really tell. So to ID the diatom, you have to clean it. And you have to clean it and remove the color from inside. And that's kind of what you'll get. This is under a scanning electron microscope, which is a special type of microscope that'll take really cool 3D images of your diatom. You cover it with, you take your sample and you cover it with gold dust, and then it will give you really high detail. So I show you this picture because it shows two views of a diatom, that basic views. So we have what's called, think of the diatom as a box. So you have your top lid and your bottom lid. So if you're looking at your Christmas box from the side, you have what's called a girdle view. And then if you look from the top, at the top of the box, then you have what's called a valve view. The girdle is just a, think of it as a rubber band that keeps the diatom pieces together. So a little bit of technical stuff, but I think this is important when you're trying to just study diatoms. So the whole shell is called a frustule. A lot of these names for diatoms are from German because the first people who described diatoms, Otto Friedrich Müller, was German. The valves are the top and bottom lids. So the epitheca is going to be your top lid and the hypotheca is going to be your bottom lid. Again, the girdle band is going to be that rubber band that holds your box together on the side. And the rafe is a long slit, long skinny opening that allows the diatom to move. So it, like a slug, I kind of like to think of it as a slug. It uses mucus and it'll release slime through that slit to move. So kind of a recap, what do they eat? So again, they're photosynthetic. They make sugar from sunlight and carbon dioxide. And they also need nutrients like plants, nitrogen, phosphorus, and potassium, but they also need silicone. Silicone's really important because, again, that's what their outer shell's made from. What eats them? So all sorts of stuff. There's some things that'll eat them by accident. There's lots of things in nature that'll eat them by accident. And then there's other things that'll eat them on purpose. So by accident, so you might have birds like ducks and other wading birds. They'll eat algae. Like, you know, if you look at a goose or a duck in the park, it'll be dabbling in the water. It's mostly going for some, maybe it's not purposely going for the diatoms, but it's eating them by accident because they're growing on the bigger algae. Turtles, again, the same thing. They're wanting to eat the plants and the bigger algae, and the diatoms will be a casualty because they're growing on the bigger algae. But other things that'll eat them on purpose are mussels. Mussels are important because they're filter feeder, and they'll eat the algae directly out of the water. And then copepods, which would be another small type of arthropod that'll eat, because they're so small, they'll eat algae as well. And then other insects like stoneflies and caddisflies. Stoneflies will eat them by accident, kind of like the birds and the reptiles. Caddisflies are what are called waste eaters. They'll grab anything they can out of the water with their net, and sometimes algae might happen to be in the random stuff they catch. So again, here's some pictures of the predators. Again, aquatic insect larvae. Here, they'll eat omnivorous. That means that they'll eat both meat and the plant material, including diatoms. But this and then mussels. And then an example of a mussel that I found in River Legacy Park. And then again, our turtle picture. So biomonitoring. Let's look at the concept of biomonitoring. So it's important because you use this idea to study stream and lake health with diatoms. So what is biomonitoring? It's using organisms such as insects, plants, and other living things to monitor water quality. Let's get back to that idea that certain animals and plants have that Goldilocks zone. They like a certain temperature. They like a certain water flow. So keeping that idea in the back of your head, let's explore it. So oftentimes when we look at biomonitoring, when we look at fish, because if you're a fisherman, you know that catfish will like certain kinds of water, while trout might like your channel catfish is going to like maybe a certain kind of water and like to be in the mud and stuff, versus your trout prefers cooler water. So that can tell you a little bit about the habitat by the fish or by the fish that you catch. Fish are great because they're easy to ID. You can use a net. They're a lot easier to catch and then release. But why would you use diatoms that you have to look under a microscope versus fish? Well, diatoms can be smaller and easier to handle than fish. I can simply go out to one of the ponds here at the one of the ponds or the Trinity River and grab a bottle of water and take it with my microscope and at least tell you, oh yeah, that's a navicula, like the one diatom I saw. Another important thing is when you're trying to look at how healthy your water is diatoms don't need special permits. When you're netting fish, you have to have a special permit through the Texas Parks and Wildlife oftentimes in order to net them and then study them. But usually with diatoms, all you need is a public space that you can access publicly or permission to be on that property owner's property. And then they also, most importantly, they respond quicker to change than fish. This has to do with a fish lifespan is longer than a diatoms. A diatoms, you might say hypothetically might live for a month, whereas a fish might live for 2 years. So when that fish was born, it might be representative of the water conditions like 2 years ago, whereas your diatom might be representative of your conditions a month ago after it's died. And also because they take in nutrients straight from the water, they have what do we call it? So since they take in nutrients straight from the water, you can tell really quickly what the conditions of the water, and they respond faster to pollution. So how do you tell if your water is dirty or clean? Sometimes we want to say that like that picture on the right with all the green, oh, that's dirty and that smells. Or that picture on the left, you want to say, oh, that's a perfect place to swim, but is it really? Looks can be deceiving. And sometimes you can't tell what's up with your water without taking a closer look. So when you're studying water, it starts by choosing a site and getting the diatoms. So here you want to, so here I need to study this river for whatever reason. So I grab a blob of algae. And from that blob of algae, now what do I do with it? I've just got a mess on my hand, right? Well, I've got to separate it now. So this blob of algae is probably green algae with on the bottom with diatoms growing on it, which is on the top. So again, plant growing on a plant. Well, algae aren't plants, but we can think of them as that. And then here comes the tricky part. So now you have to identify or tell your diatoms apart to species. And that's tricky whenever you have stuff inside the shell, because the problem with the shell is see-through. And if you have stuff inside your Christmas box, you can't see, you might not be able to see what the box itself looks like when you're looking at a bright light. So you have to go through a very caustic process. You have to clean the diatom and then put it on the slide. And then on the right, this is what you get. So let's take a kind of a look at this. So it's long and it's skinny, and you have all these little lines or ribs going down it. You're not able to see that clearly until you clean out the trash on the inside. It's not trash, but I guess when you're trying to ID your diatom, it could be thought of. So what are we looking for when we're trying to identify the species? Well, under a microscope, it comes down to shapes and details. So I like these two examples. The one on the right is round, big, and flat. This is a genus called Cochineus. And then the one on the left is long, skinny, and wavy. I think these guys are cool. This is a genus called Eunotia. Cochineus on the right grows on flat surfaces, and Eunotia will grow kind of like a flower, growing from the skinny end and growing out. So for biomonitoring, how are diatoms used? Well, let's revisit that idea of, let's look at this diatom right here. We're going to name him Gomphy, for short for Gomphanema. So Gomphy doesn't like it super cold, and he doesn't like it super hot. He likes it in the Goldilocks zone, just right in the middle. So he's probably going to want to live in water that's in just right zone. So again, that goes back to habitat preference or where they like to live. But if Gomphy disappears over time, then that might give you a clue, did the water temperature change because Gomphy is no longer there. So we can use, by recording the diatoms or the species we see over time, we can get a good idea of how the habitat of how the water has changed itself over time too, by using them as a proxy. And that's where it comes into tracking environmental change. You take enough diatom samples over time and you ID enough from enough seasons, then you can see, well, Gomphy was here and then he went away these years. He was here like in 2001 and then he disappeared in 2005, but then he came back in 2010. Then you know something's up with the water temperature, because he only likes it in the middle. So habitat preference. Species have habitat preferences like water temperature. The species found gives clues to conditions such as temperature, growing surfaces that are present in the water body. So this is a case to where Gomphy is not happy on the right now. It is too hot. He will say adios, and he's not going to be found in your water. However, now we have this diatom, I'm going to call her Tabby, short for Tabellaria. Tabby has entered the scene now because she likes it hot in the water. So when you don't see Gomphy anymore, but you see Tabby, then you know that the water temperature has probably increased from that year or that season. So habitat preference and biomonitoring. So this is just a graph. I mean, this is just a graph that kind of shows you temperature. So on the left, we've got growth rate, just how many like the diatom population. And then on the bottom is temperature. So if you see to the left, the graphs, you know, they have the humps on the far left where it's colder. That means that those diatoms like it where it's cold. And then we have the line on the right, the hill on the right, where it peaks at about 15 or 25, that means that diatom likes it hot. So if you see these diatoms on the left, you know it's going to be cold. If you see this diatom on the right and only this one, then you know it's probably going to be hot because they have different habitats that they like or conditions. And that's how you kind of tell it graphically. So let's go into the topic of algal blooms now. Algal blooms are when you have like a sudden explosion of one species of diatom or algae. Think of red tide in the ocean. Out of nowhere, you have this red algae that explodes and it kills everything off, and all you'll find is that red algae for the red tide. So it's kind of a boom and bust cycle. So you'll boom with, yay, it's a party, all of us red algae or diatoms, and then bust and they'll all disappear and leave the party. So what causes them? There's different causes. So one common cause would be like runoff from fertilizers. So it's like, think of that farm or that factory that's throwing in all the nitrogen and all the extra nutrients. It's like throwing in all the cookies instead of the salads. You're going to have all those diatoms coming in for the cookies, and you're just going to have an explosion because everyone wants cookies. Another cause could be, and because you have cookies instead of salad, everything's out of balance. So that could be like a form of pollution whenever the system is out of balance. What is another cause? So another cause would be hot and dry spells. Hot and dry spells can be bad because it's not the normal conditions in the water, right? So one, we remember that heat speeds up algae growth. So all of a sudden it's cold and it turns hot. You know, your tomatoes this time of year after a certain date, it's like, whoa, where did all that new growth come from? Same deal. It's because of the warmer days. And then another thing that can cause algae blooms is when it's dry. So hot, dry spells, what we often see in Texas. The dried out water bodies concentrate or collect all that food in one place. And so the combination of all the food in one place and the warmer temperatures with all the algae to eat it causes the bloom. And then another cause of algal blooms is when you simply have nothing that will eat you. In this case, invasive species. We all know Nandina and we all know privet that we were talking about earlier. They explode here in population because there's nothing that eats them. Well, same thing with this algae called rock snot. And if there's, for the participants who are here, the microscope image that I have over there, that is rock snot under the microscope. So general symptoms of algal blooms. So water kind of comes in layers. So the bottom layer, middle layer, and then the top layer. So one symptom, there's very high oxygen at the surface because all the algae floats to the top. You'll have way too much oxygen at the top, but because everything dead floats to the bottom, that eats your oxygen, and that's what causes fish kills, because there's no more air for the fish to breathe on the bottom. It also raises the water pH, or makes it more like not bleach, but puts it on the same, makes it more alkaline like bleach. And that's because carbon dioxide actually makes the water more acid like vinegar. And the algae are using up all the CO2. So now you don't have anything to add acid to it. It becomes a base. Other symptoms are an off color. When you look under a microscope, only one algae present, and then you might come with fish kills. This particular picture, I imagine, looks to me like blue-green algae, maybe microcystis. If you've ever gone to a pond or stock tank in the summer and it smells like sewage and it's green like that, it's probably microcystis, which is a totally different type of algae. So let's get to our friend Didymosphaenia geminata, or Didymo, or rock snot. So this algae is native somewhere, but it's not native here. And it's certainly not typically native in Oklahoma, where some of this is a problem. So this is a large algae. Typically it grows in strings. And it's really pretty under the microscope. It almost looks like a bottle or a mummy. And its habitat is in cold places. And in the right places, there's stuff that'll eat it and keep it in check. Well, the problem comes in when you don't have anything to eat it. And that happens. So it is called, so remember that diatoms create mucus to move around and to attach to things. What happens when you have a lot of mucus? It feels like snot or boogers. So known as rock snot due to its stringy and mucousy texture, this algae doesn't occur in Texas. But I bring it up because this is a good example of an invasive diatom in the US. How it got here, we don't know exactly, but it started up north and it kind of worked its way down. The reason why, so in Oklahoma, it's found in the Broken Bow area where you have where you might find cold water species like trout, and it's particularly found where there's the cool water dam releases just downstream from the dams, because it likes that cool flowing water, and it becomes a problem because one, it's obviously a trip hazard. You slip on this stuff. But also because you have a monoculture of Didymo, it literally smothers out your water insects and larvae. So bye-bye dragonflies. It kills your fish, goodbye catfish and trout. And you're just left with this mess. And also, who wants to go play in this? Like, so if you went to Chickasha National Recreational Center and you saw this, would you be happy? So to get on the lighter note, let's go on just a gallery of some of the diatoms you might find in Central Texas. A little bit of background about these. So I did my thesis on part of it on the Colorado River in Central Texas. And these are some of the samples I took from grad school. So this is Cyclostephanos dubius. We've got a top view. This is one of your, and a side view. This is one of your smallest species. It's planktonic. Cycloteleminigiana. This is a little bit bigger. This is a really common one. It also floats. What's cool about these circular guys is that the circle shape helps it float, helps these diatoms float. Because of the increased surface area. It's kind of like a feather, you know, it has bigger area to help it float in the air, just like these diatoms do. This is a really cool one. It looks like a star. So Stephanodiscus neoastria. It's called neoastria, astria for star, because it looks like a star. Again, this is another floating species. And then this sucker, I hated him because he is really hard to, even under the highest power microscope, to identify. He is so the size of a human hair is about 70 microns. Trying to think of how big that was. So you know how small, like if you look at a human hair, it can be. So this guy, you would probably need about at least 20 or more of these guys side by side to even get halfway on the width of a human hair. They're that small. So now we go on to some, so those are called centric diatoms or circular diatoms. I'm sorry, this is, every time I saw this genus, I thought of Lord of the Rings and Boromir.
You don't have to apologize.
So this species is not that common, but I think it looks cool. It's a moving species and it moves in the sand. If you've ever been into like Colorado Bend State Park, you know, some of it's kind of muddy, that fine sand. That's where that guy likes to live. Diplonaea pseudovallis. Pseudovallis means fake egg or fake oval. And it comes in different, and I like this because it shows you all the differences on the same species. So we got a tiny guy based off of maybe tiny guy and then big guy in the middle, and then a smaller guy. And you can see even within the same species, there's a lot of variation. Navicula cryptocephala. Cryptocephala is Latin for hidden head. This one is kind of a diamond shape. This was one of the more common ones too, and this is kind of tells you about the conditions of the Colorado River again, because it has that sandy bottom. This species also likes it where it has the water's more salty. So again, gives you a clue of what the water conditions. Nichia. So this one's a really, and I show you this because it shows you the diversity even within one genus, the diatoms. So on the far left is Inpalia, which was the most common one, and then we have Insinuata, and then just some random ones to show you how tiny to how big they get. And these are all size to scale, about to scale. And this one wasn't super common, but again, Nichia curziana. Again, like salty condition, like sand, and he likes salty water. So you wouldn't have thought, when I say salty, high mineral content. So again, it tells you a little bit about the Colorado River. If we're seeing some of these floating species, but also species that like sand or more salty mineral conditions. And then a Nichia amphibia. I just threw this one in because it's cool. You can tell really well. In order to ID some of these diatoms from one another, you literally have to count the ribs on them to tell them apart. So you have to tell how many ribs per inch kind of deal. And try doing that 1,000 times to ID stuff. And then Sterorella. This one's cool. So we have Swissika and Brevissonii. These are actually potato chip shaped, which is super cool. So if you think of a saddle, the saddle, if you think in your mind, it turns, it helps it twist into the sand like a screw. So if I piqued your interest on diatoms, I do have some recommendations of books. That middle reference, Diatoms in North America, or diatoms.org. It's completely free and has more information than you can want on diatoms. So check it out. Also, TCEQ has on that third line has a free intro to freshwater ecology. If you're just interested in water and streams in general, I'd highly recommend that book. And also that top book I have a copy of over there, which is the green cover. Any questions or comments?
I have a question from chat.
Okay.
First of all, an early comment on that early slide you had where they were all arranged, and artistically, was they are beautiful. And yes, I agree, they are beautiful. When you were talking about the shells and they reproduce, that piqued this question. Is each shell unique like ice crystals or like snow? I think what you meant to say was like snowflakes. Or are the shells pretty much identical?
The shells are almost identical, but you have to think it's like the shells are almost going to be identical, but the shell shape might change slightly if the water has more salt or other minerals. That'll actually affect how the shell forms, if it'll have more holes or less holes. So that can be another clue of water quality as well, depending on the species.
I just realized they might not hear me asking the question, so I can easily get louder.
I think we had a question back there.
Go ahead.
Both.
Okay, repeat the question kind of.
So she asked, are diatoms plankton or do they grow on plankton? They are plankton because they float in the water. So they're part of that, they're part of the green matter that grows in the water, but they also grow on plankton. So you could technically, if you had a piece of floating big algae, then the diatom could be grown on the bigger algae.
Yes. Okay. Oh, I'm not sure. I should stay away from everything. My recollection, and I don't, if someone, it says I'm wrong, I accept it. My recollection is that anything is plankton if it's moving because of the wind or if it's moving slowly. It can't move any faster than the current. So even jellyfish are plankton. Does that sound right?
Usually when we think of plankton, they're you can only see them under a microscope or they're really tiny. Ah, gotcha. So I'm with okay.
Figure out where the cells are going from like from to like the diatomaceous earth.
So diatomaceous earth is actually a fossil. So your diatomaceous earth is going to be diatoms that are millions of years old. So they get buried in the sand. So you have your sand, say you have your sand that floats to the bottom, and then that gets buried. It floats to the bottom, and then that gets buried. And so that's all the diatoms are. They floated to the bottom at some point and got covered by something else.
And so they call them, they locate them.
Good question. All I know is just like you might find limestone in one place and granite in another. So I guess it depends on the geology. Or I'll go ahead and front row first.
Thank you. I was curious about the gold dust. You mentioned gold dust that you use to be able to actually clean them.
So
To take a scan.
yeah.
Yeah.
So I don't know all the particulars of the SEM or the scanning electron microscope, but I know that they coat them with gold dust because something about it allows the equipment to capture the surface better.
Water, H2, it only has two, well, H only has one electron, oxygen has 8. So the electron beam would go straight through water.
Okay. This is a real stupid question. What makes them a crystal?
So a lot of people, so I would say technically they're not a crystal, but people describe them as crystals because of how they look. So if you look under this, doesn't it look like a crystal? People also call them crystals because they're an inorganic material, silicon or silica, instead of something organic like other types of algae.
You were talking about rocks getting a slimy cover.
Mm-hmm.
So they're all slimy rocks and diatoms.
Not necessarily, but it would be a clue that they're probably diatoms there. It's not always going to be, it could be some other type of algae or something else putting that slime there, but it usually diatoms.
Study. As an indicator of what's going on in the water, or in other words, like you have the prevalence of a bad diatoms, you have other diatoms that is stuff about.
That's a good question. It depends on your focus. So some people are interested in you might want to mute your microphone. Okay, let's try this again. So it just depends on your study focus. Usually as an ecologist, or the ecologist in me, we don't necessarily, I wouldn't want to introduce anything that isn't already there. because we've seen what happens with invasive species. We think it's not going to reproduce, but it does.
So it's just pretty much tells you what's going, you from find out what's going on.
Yeah,
Figure what that-
right.
On third grade science levels.
Yeah.
You said that it starts the food team and the food web,
Mm.
but is it considered a producer?
Yes, a primary producer. So your first producer.
But not
So-
a-
It's not a plant, but it's a primary producer like a plant. What's interesting, when the diatoms were first discovered, because they move, some of them move, the researchers thought they were actually animals, and they were first categorized, grouped as animals before they later learned that the algae was different than animals.
I stand collected jellyfish are surprising, and I have a degree in biology.
Oh.
There are many marine diets,
Yeah.
many, I mean, there's some diatoms, there's tons of diatoms. Is the diversity among marine diatoms, is it greater for marine diatoms?
Yeah, so marine diatoms are going to be more diverse because, well, one, that's where diatoms first
Yes.
evolved, was in the ocean. But your greatest diatom diversity is actually at the poles.
Oh, no way.
So what's weird is that you think most diversity goes is biggest at the tropics and the smallest at the poles. Diatoms are the reverse. They do best in the cooler water, but that has to do with because there's more. It goes back to air. More colder water can dissolve more carbon dioxide in air, and that has to do with it.
Or krill.
Yeah.
Oh.
Yeah.
I have a question from online again, and it has to do, it's sort of multiple.
Oh, sure.
This will be your experience.
Okay.
So just how do you collect them? And I think you answered this before, but you said a straw. But then do you take your microscope to the field, or do you take your water samples back to the lab?
So typically you'll collect out in the field and then take it back to the lab. How many weeks did it take? So if you really want to study the shells, you have to clean them out. So you use a multi-step process with hydrogen peroxide, and I believe I used hydrochloric acid. And so you want to have access to a fume hood. There's different ways to clean them out, but that's a more common way. So it's kind of lots of not nice chemicals.
I'm just thinking, I would have lost them. I would have dripped some hydrogen peroxide on that sample, and it would be gone.
Yeah, so you have to be careful not to contaminate. What's funny is that during a story, so kind of just a mini story, there was a guy in the diatom community back when Russia was the Soviet Union, so the closed borders. Well, this diatom researcher wanted to take samples from Russia back to the US. Well, the problem is you can't take water samples even in the 80s. So what he ended up doing was taking business cards and drawing the samples into dust and then folding them up and then taking them overseas in his business cards with him and then rehydrating them and mounting them.
Cool.
So there's ways to kind of illegally get stuff across customs. I guess in the back.
So, when you clean these things up, you basically kill the living, right?
Yeah.
Okay, that's the delegate.
Again, the question is when you clean them, do you kill the living part?
Yes, you do. You have to empty the Christmas box in order to see what the outside of the box looks like because it's clear.
Okay, so then you're looking at the shell that this not plant, not animal has put in.
Yeah. So let's go back to that one slide where I showed the two of them.
Phytoplankton, so they're.
Yeah, specifically.
But they're not in kingdom planting. You know, she's younger than we are, and I learned 3 kingdoms in high school, and then some people say there's 17 kingdoms. which is ridiculous.
The only thing they have in common with us is that they have a nucleus, I would say.
Feel free to say that's way too advanced, but we had a question, and you say they sexually reproduce. We think of pollen, we think of pollen, right, and a plant ovary. So, or is it simpler than that, or is it like super advanced, you know?
So what you'll have is, from what I understand about sexual reproduction, you'll have two diatom cells before they get together. They get rid of their shells, they dissolve them. And so you'll have what are called gametes, or the sex cells, that'll fuse together into a new diatom. And then after, it's naked, so it has to grow itself a new shell, but it's bigger too. It has its birthday soon.
Future trends of biomonitoring and climate change. Would be clear the future trends there.
The question is about future trends, biomonitoring, climate change.
Oh, that's an interesting. So there's starting to be. So traditionally, we've used things like microscopes and physical things. So just physically looking at, okay, this is the shape of the diatom, this is a species. Well, that's a problem, especially with small things like this, is because there's things called cryptic species, or species that are hidden. That happens whenever you have two or three diatoms that look so much alike that you can't tell them apart at the species level. You can't tell Jane from Joe. So that's where DNA comes in. You can tell Jane from Joe apart by looking at their DNA, because it'll actually be, though they look the same on the outside, their DNA is going to be totally different. And so a new trend is what's called eDNA, or environmental DNA. You'll take a sample of your water, and our technology has gotten so good to where there'll be traces of DNA left over from, say, the fish or the algae or the plants grown in there. And so you can take those chunks of DNA that are floating from the environment or your water, and you can tell what is in your water by just taking your sample and what's floating in it. And so to answer your question, that's a new thing that they're trying out, environmental DNA, the DNA that's floating and left over from the diatoms. Because it's a lot more accurate whenever you can tell, you get a more accurate picture or better picture of your species when you get the DNA that's easy to tell apart, versus the two shells that might look the same to the human eye.
Are they harmful to people? Swimmers. I assume that means people.
I would say generally not. It's like my view with any other living thing, that's neither really good or bad. It just depends on the environment it's grown in or the habitat. Take rock snot or dynamo. It's in its natural environment, like that sample right there is from the Arctic Circle. It doesn't overgrow, it's in its happy place. But if you put it in Oklahoma, downstream of the dams, it's going to take over. So in that case, it's quote unquote bad.
I said that I'm trying for this.com, there's only the world.
Yeah.
Now, how do you get out of Silicon Valley? Did I split it in the middle?
So
The premise.
he was asking about, in my introduction, that diatoms produce 20 to 30% of the oxygen in the world, especially from the oceans. How does the air escape from the diatom shell? So diatom shells aren't, they have holes in them. So they have holes to get air and also to get food.
Any more questions? We have asked a lot. This is really good.
I'm glad to.
Sometimes it's like,
It was more about these spores and humans.
Yeah.
Like that picture of the brown one, that Victorian slide, that they ground in the little. So are each slit, where it looks like there's a center, and it looks like it's a radiating pattern of slits. Is each one of those radiating slits of perforation and natural perforation?
So just to note these, she was asking, are all those details in the diatoms, are all those holes? Just to let you know about these pictures, these are many different diatoms arranged in like different, they're many different diatom shells put together and arranged in a picture. But that center one is a circular one. Those waves are not openings. They're just waves. They're ribs. Think of them as a rib on top of the circle, on top of a dinner plate. And that's what that detail is.
Yeah, I know there's a theory going on, silicon-based life, would that translate these, they have silicon around?
No, so silicon-based life is based off of so we're carbon-based. And our DNA. Yeah. So when they say carbon-based, our DNA is based off of carbon. When they say silicon-based life forms, it's based off of silicon instead on their DNA.
Some very old Star Trek episodes.
Yeah, she did great. Woo! Thank you so much. Thank you for that.
I know it's not easy, and then you got bombarded with questions, but
She's like, yeah. No, sir.
you're famous now. Yes, and I even will remember your name, maybe. Breena, that was Breena Riley, class of 2022, one of our very own. Does anyone here in person have any announcements? Okay. I'd like you to come up here and talk to the microphone so the folks at home can hear you.
I'm just reminding you, I'm bad about putting it in the newsletter, but the Mansfield Monarch Festival, I wanted to have a Cross Timbers Master Naturalist tie-in with that. We're looking to have a Master Naturalist station, maybe a pond station, a journaling station, a tree discovering station. Let's see here. And then maybe either insects or birds. The city of Mansfield does ask that you sign up to do a volunteering on their site if you haven't done that before for a one-time code. I've got my phone number, I've got papers, and I will give it to her to send out at a blast. Oh, sorry. It is going to be May 13th. It's from 9 to 12. And again, it's the Oliver Nature Park, which is located in Mansfield off of Matlock. And anyway, my phone number, I'll give it out to you all as well. So, okay.
I just wanted to let everyone know that the Native Plant Society in North Central Chapter is having a plant sale on May the 6th at River Legacy. And we'll have beautiful plants. We don't have the plants online yet, but they will be there soon. If you go to textnativeplants.org and you can see them all. All right, thank you. Everybody come to the sale.
txnativeplants.org. Yeah. Zach. Yeah. And then Gina. You're getting lots of applause and And
So you all know me, I'm Zach Chapman. I am the current Fort Worth Lights Out program intern, and we need volunteers to help us get up and survey dead birds. We get up at 4:30. Well, I get up at 4:30 in the morning. We have to be there at 5:30 at the TCC Trinity River campus, and we need more volunteers. We are picking up a lot of good stuff. So yes. It's happening right now. So from April 1st to I think May 27th, I believe is whenever it cuts off. I think they will have it picked up for that as well. I'm just a spring intern.
I believe if you got the newsletter, I believe the links that you would click on and I know Madison put a big long article in the newsletter. So get past my boring stuff to Madison's article and it'll tell you, they'll even help you find out where to park and what the dates are and how to sign up.
Yeah, it's kind of complicated. We all get turned around when we're trying to park at this other thing, but yeah, we need people to come out and just go
Are you doing it every day of the week?
I'm doing it. Well, I'm doing it Monday and Wednesdays, but they are doing it most of the week. So even some Saturdays, I think, but mostly it's Monday through Friday. About like we get done like 8 or so, but it kind of depends on if we want to do like we have like 10 buildings that we survey, but then we also have like a few that are not on that 10, and then we just go around those a few times and see what's around.
Questions. One. Are you allowed to, for example, would you, like, I have school, still doing this, get out at 3:20, could we be here on the university?
It's in the morning.
Yeah, it's in the morning.
Okay. After it's okay.
Okay, that was one question. The other two are moved. Okay. Thank you. We're excited to
have Zach be an intern. Yes, absolutely, absolutely. I believe we're done with the business meeting. So I guess I'm calling the end of the business meeting. I think I have some numbers to call for the it's not a raffle. And okay, one more, but you get that ready, Joseph.
I wanted to thank Bill, Karen, Brent, Michelle, Melinda,
Sandy.
who else is there?
Sandy was there.
Sandy. For the Bio Blitz at Glenwood Park, I felt like there was a little, maybe not quite as many kids as we were hoping for, but there were some kids there, one TCU student there, a lot of interest and a lot of exploration. And so I think it's a lot of good momentum. And then this Saturday, there's a cleanup, and I know it was in the newsletter. So if anybody's interested, just come on over.
Thank you. Thank you. There's several events on Saturday. Yay, Earth Day. Glenwood Park, Fort Worth Nature Center. I should probably name that 5 times because there's a lot going on there. River Legacy Park, and you may know of some other areas that are doing Earth Day. So, all right. So at this point, then I'm going to close the meeting, and you can stop recording, Nancy. Thank Nancy Curl for being our Zoomer